Welcome to this lecture on human physiology, specifically focusing on male and female reproduction. Today, we are going to explore the incredible biological machinery that makes the creation of human life possible. We will look at how the male and female systems operate using completely different strategies, one acting like a continuous, high-volume factory, and the other like a highly precise monthly biological clock. By the end of this session, you will not only understand the physiology behind these systems, but you will also learn how to use simple mathematical models to evaluate fertility, predict ovulation, and monitor the health of an early pregnancy. Let us dive right in. To begin our journey, let us look at the male reproductive system, which operates exactly like a highly efficient, non-stop manufacturing plant. Why does it need to be a continuous factory? Because the journey a single sperm must take to fertilize an egg is incredibly hostile and physically demanding. To overcome these harsh odds, the male body relies on sheer volume. The process of creating these microscopic genetic packages is called spermatogenesis, and it takes place inside the tightly coiled seminiferous tubules of the testes. This intricate process is heavily regulated by two key brain hormones: luteinizing hormone, which stimulates testosterone production, and follicle-stimulating hormone, which nourishes the developing cells. The entire cycle takes about 64 to 72 days to complete. Now that we understand the continuous nature of this biological factory, how do we actually measure its efficiency? In reproductive medicine, doctors evaluate the functional success of spermatogenesis by calculating the total motile sperm count. This formula tells us the true fertile potential of the male system. M total represents the absolute number of moving sperm in a single sample, measured in millions. To find this, we multiply V, the total volume of the fluid in milliliters, by C, the concentration of sperm per milliliter. Finally, we multiply by P motile, which is the fraction of those sperm that are actively swimming, expressed as a decimal. If either the volume or the concentration decreases, the total motile count drops significantly, which directly lowers the probability of natural conception. Let us apply this formula to a real-world clinical scenario. A patient visits a fertility clinic, and his laboratory results show a total volume of 3 milliliters and a sperm concentration of 40 million per milliliter. The technician notes that 40% of the sperm are actively swimming. We need to calculate the total motile sperm count. First, we multiply the volume of 3 by the concentration of 40 to get 120 million total sperm. Next, we multiply that massive number by the motility fraction of 0.40. This crucial step filters out the non-moving cells, leaving us with only the viable swimming cells. The final result is 48 million actively swimming sperm. Since a count above 20 million is considered healthy, this patient's reproductive factory is functioning exceptionally well. Having calculated the output of the male reproductive system, let us visualize exactly where this mass production takes place. What you are looking at is a cross-section of a seminiferous tubule inside the testes. Notice how the process moves from the outer edge toward the hollow center, or lumen. At the very outer boundary, we have the primordial stem cells, called spermatogonia. As they divide and mature, they move inward, transforming into primary and secondary spermatocytes, then spermatids, and finally fully mature spermatozoa with visible tails extending into the center. You can also see the large Sertoli cells that nourish them, and the Leydig cells in the space outside the tubule that produce testosterone. This diagram perfectly captures the continuous, layered manufacturing process we just discussed. Now, let us shift our focus from the continuous factory of the male to the highly orchestrated cyclical theater production of the female reproductive system. Imagine a grand stage play that takes exactly 1 month to prepare, culminating in a single spectacular main event, the release of an egg. Unlike males, who produce millions of sperm daily, females typically mature and release just one single egg per month. This requires an incredibly precise biological clock. The cycle is divided into two main parts: the follicular phase, where the egg matures, and the luteal phase, which occurs after the egg is released. The release itself, called ovulation, is triggered by a massive, sudden surge in luteinizing hormone. Because the released egg only survives for 12 to 24 hours, understanding this exact timing is absolutely vital. Because the window of opportunity for fertilization is so incredibly narrow, we need a way to mathematically predict this critical moment. While the total length of a menstrual cycle can vary wildly from woman to woman, the second half of the cycle, known as the luteal phase, is biologically fixed at approximately 14 days. This is due to the fixed lifespan of the corpus luteum. We can use this biological constant to predict the day of ovulation, denoted as D-ov. To find it, we take L cycle, which represents the total length of the woman's menstrual cycle in days, and simply subtract 14. If the total cycle length increases, the day of ovulation is pushed later into the month because it is only the first half of the cycle that varies. Let us see how this simple subtraction empowers patients with actionable medical knowledge. A female patient is trying to conceive and notes that her menstrual cycle is consistently 32 days long. She wants to calculate the exact day she will ovulate to identify her most fertile window. We take her total cycle length of 32 and subtract the biological constant of 14 days. This backward calculation isolates the duration of the variable first half of her cycle. The result tells us that ovulation will occur exactly on day 18 of her cycle. Furthermore, because sperm can survive inside the female reproductive tract for up to 5 days, her most fertile window will actually begin on day 13 and end on day 18. This precise timing perfectly complements the continuous sperm production of the male system. To truly grasp how this biological clock operates, let us look at the hormonal fluctuations that drive it. This graph tracks the 4 major reproductive hormones over a standard cycle. On the horizontal axis, we have the days of the cycle. Notice the massive, sharp spike in the red line right in the middle. That is the luteinizing hormone surge that directly triggers ovulation. Before this spike, during the follicular phase, you can see estrogen rising as the egg matures. After ovulation, the ruptured follicle becomes the corpus luteum, which produces the large wave of progesterone you see in the second half of the graph. This shaded region is the luteal phase, and as we just learned, its duration is strictly fixed at 14 days, which is the cornerstone of natural family planning. Now that we have explored both male and female systems, what happens when they successfully interact? Imagine you have just planted a seed in a garden, and the moment it takes root, it starts sending out a loud radio signal to keep watering it. When a fertilized egg implants into the uterus, it acts exactly like this signaling seed. The female body needs an immediate, powerful signal to stop the normal menstrual cycle and prevent the uterine lining from shedding. Almost immediately, the outer layer of the embryo begins secreting a hormone called human chorionic gonadotropin, or HCG. This hormone rescues the corpus luteum, ensuring it continues to produce progesterone. To ensure survival, the concentration of HCG in the mother's blood grows exponentially, doubling every 2 to 3 days. To understand why this rapid multiplication is so critical, we must look at how we model its growth. If HCG does not rise fast enough, the pregnancy will be lost. We can model this rapid multiplication using an exponential growth function. In this equation, H of T represents the concentration of the hormone at a specific time. H0 is the initial concentration of the hormone at our first measurement. We multiply this initial value by a growth factor of 2, raised to the power of T divided by D. Here, T represents the elapsed time in days, and D represents the doubling time, which is the number of days it takes for the hormone to multiply by 2. If the doubling time increases, it means the hormone is growing slower, which is a major clinical warning sign for a failing or ectopic pregnancy. Let us use this mathematical model to monitor a very early pregnancy. A patient's initial blood draw shows an HCG concentration of 100 units. The doctor wants her to return in exactly 6 days. In a healthy pregnancy, the doubling time is typically 2 days. We need to calculate the expected hormone concentration. We substitute our initial concentration of 100, our elapsed time of 6 days, and our doubling time of 2 days into the formula. The exponent fraction, 6 divided by 2, simplifies to 3, meaning the hormone will double exactly 3 times during the waiting period. 2 to the third power evaluates to 8, which is our total exponential growth multiplier. Multiplying 100 by 8 gives us a final expectation of 800 units. If her actual test result is significantly lower, the doctor will immediately investigate for complications. To fully appreciate the sheer speed of this biological signaling, let us look at the exponential growth curve of HCG. On the horizontal axis, we have the days after implantation, and on the vertical axis, the hormone concentration. Notice how the curve starts off relatively flat, but quickly becomes incredibly steep. Starting at 100 units on day 0, it doubles to 200 on day 2, 400 on day 4, and hits 800 by day 6. This rapid, upward curving trajectory is the hallmark of exponential growth. By using this visual and mathematical baseline, doctors can accurately predict healthy hormone levels and quickly identify failing pregnancies. This incredible signaling system bridges the gap between initial fertilization and the long-term development of the fetus. We have covered a tremendous amount of ground today, so let us review our key takeaways. First, we learned that the male reproductive system operates as a continuous, high-volume factory, and we can measure its success using the total motile sperm count. Second, we saw that the female system is a highly precise monthly clock. Because the luteal phase is biologically fixed at 14 days, we can mathematically predict the exact day of ovulation. Finally, we discovered that early pregnancy relies on a rapid, powerful chemical signal called HCG, which grows exponentially to halt the menstrual cycle and protect the embryo. You now have a solid understanding of both the biological machinery and the mathematical models that govern human reproduction. Excellent work today.